Okay, so this is for your Microscope Lab. You have a Microscope Lab worksheet where you have to identify parts and functions. And then I'm going to show you how to make a wet mount slide, how to look at a preserved slide. You're going to have to draw the images. Those will be separate photos that I'll download for you on Rock. And so you need to look at those photos and draw them in on your lab. Okay, first of all, this is a compound light microscope. This can magnify, you can buy ones that magnify up to 2,000 times. This one can magnify up to 400 times. Now you need to know how to find total magnification. The eyepiece always is 10x, meaning it magnifies something 10 times the normal size. And then you have objective lenses. The short one here is a 4x. Then the next one locked in place is a 10x. And the last one is a 40x. So when the 40x is down, the 40 times, the total magnification is 40 times 10. So 40 times 10 is 400 times. If I went back, just simple math, here's the 10x. So now 10 times 10 is 100. So if you had something that was an inch long, it would be 100 inches as far as what you see, 100 times the normal size. So microscopes are used to see things that are small, smaller than millimeters. So the parts of the microscope, the part you look through with the lens here that magnifies 10x, and both lenses do, it's just 10x. These are called the eyepiece or the oculars. Then light will go through, comes through this part here. It's usually called the body tube. There's a grip here, and there's a grip on the base in the front because you're supposed to carry them with two hands. Okay, this is called the arm, and this is called the base. Now what I do is that this is called a mechanical stage. This is a mechanical stage clip to hold the slide in place. I'm going to take this slide, it's the letter E. It's already been made, it's a small case E. And I'm going to place it so the E is right side up. I'm going to push it to the back. I'm going to make sure my light is on. This is the switch for the light. This is called the light intensity dial. And then you'll see this, which is called the mechanical stage knob. And I'm going to move that knob and look from the side here until that E is lined up where the light's coming through it. And I'm going to start on the 4x, the smallest objective. This is called the scanning objective. The 10x is the low power objective. And the 40x is the high power objective. If you go on to take microbiology, there'll be a fourth objective that will be 100x. Then I'm going to move this bigger knob that is called the course adjustment knob. And you can see when I move it, there's one on each side. When I move it, the stage moves. As soon as I get my slide in, I'm going to take that course adjustment knob and I'm going to move the stage all the way up. Then I'm going to look through my oculars, and I may have to reposition my slide so I can kind of see the E, it's blurry. Then I'm going to take my fine adjustment knob and I'm just going to move it until I can get a clear image. If that doesn't work, then I'll just use my course adjustment, which is what I'm doing, to adjust it so I can get the letter E perfectly. There it is. Now the letter E does not look the same because lenses bend light. Like if I look at an image like a clock, I'm looking at a clock, and if I could see that clock in the back of my eye, it'd be upside down. You have two lenses here. You have an objective lens and you have an eyepiece lens. So light is flipping the image twice. So that E should appear upside down and backwards. And if you look at the image, at the photo that I'll upload, you should be able to see that. Once I have it focused in, I take it and I move this. This piece that moves is called, and I gotta lock it in place here. I'm trying to think if I should give you the full name or not, because people always do the short name here. It'll be easier to do. Can you stop it? Oh, I can't pause. Anyway, I'll show you, and then we'll go back to that. So I focus in on this with the low power objective, and then I'm going to turn it and lock it in place. And once I've locked it in place, the image will be bigger. And I'm going to take this fine adjustment and I'm going to move it until it becomes crystal clear. And now that E is pretty much taking up my whole, what we call the field of view. And now I'm going to move this one more time. And I'm going to move my objectives and I'm going to lock it. It's going to hit, and it does hit, but there's a little spring in there. So I'm going to lock it in place, and now I can't even see the E. All I see is the ink. Now you should never ever, when you're on this high power objective, you should never ever touch that coarse adjustment knob, because if you do, you can crack the slide. You always want to use the fine adjustment knob here to lock it in place. When you are done looking at the slide, you want to move it back to the scanning objective, and then you want to you can either take the slide out right now, I don't get too picky about that, or take your coarse adjustment knob and take the stage all the way down. Okay? And once you've done that, and you're on the scanning objective, you could put your microscope away once you turn off the light and unplug it. This is your light source down here. There's also what we call a condenser. I don't get too excited about you knowing the condenser. There is a little structure, it's hard to see, but it moves back and forth. And what this does is it changes how much light is coming through. There's two kinds of diaphragms. Diaphragms, both of them. There's one down here that you can move that makes more light come in, or I always just like to use this little lever, this diaphragm lever, and that will change the amount of light coming through. So there's a lot of different ways to let different amounts of light go in through your slide. What was I going to say? Anyway, I'm going to show you how to make a wet mount slide real quick. So you just take a slide.
Do you want this separate?
No, this is fine. And a cover slip. And you're going to put some iodine on, because iodine helps to stain. You can use all kinds of stains, methylene blue. I'll put a drop of iodine in there. And then you probably did this in high school. You take a toothpick and you gently scrape the inside of your cheeks. Now when you pull the toothpick out, you shouldn't have like a chunk of cheek on there or blood. Cheek cells are sloughed off all the time. And then you simply take it and you let those cheek cells go loose in the liquid, and then you just drop your cover slip on. Now, when I drop the cover slip on here, I got a bunch of bubbles. So I'm going to just take that same toothpick and gently try to get them out of here. Boy, I've never got so many bubbles ever. Because bubbles will obscure your ability to see some of those cheek cells. So I'm going to move this around and get those bubbles out of there. Come on. Because I'll take a picture of this too, and I will load it on there so that you can see cheek cells. And you can anticipate that the letter E and cheek cells will probably be on your lab exam. And I'm gonna stop here just so I don't want to grab something. Okay. So I'm going to take now my slide, I'm going to open up my stage clips, I'm going to put that in there, and I'm going to move it so that I can see the light coming up through it. I'm going to start on my low power objective. I'm going to focus in, so I'm going to bring the stage all the way up and then bring it down to see if I can see any cheek cells. Now the cheek cells are tiny, but there's some right there. And so I'm going to change the light with my diaphragm. I'm going to let less light in because the cheek cells show up better. Then I'm going to move to my next objective. I'm going to lock it in place. And now I can see my cheek cells even better. I can see the cell membrane, I can see the nucleus, I can see the cytoplasm, and then I'm gonna go to the next higher. I'll lock it in. There they are, those are nice cheek cells. And again, I'll take a picture of these and I will load them so that you can draw them and label the parts on your Microscope Lab. And then again, when I'm done, I go back to the low power objective. I move this down, the stage down. I take my slide out. And if you were in lab, you'd have to clean this off, especially nowadays. Anyway, so those are the parts of the microscope, and I hope that helped. You'll want to go through, there's a matching part on your Microscope Lab. I'm also going to give you a separate microscope sheet for you to label. And again, you'll need to pay attention when things are due and make sure you get them in the drop boxes by 11:59 p.m. on the due date. Okay, that's it for the microscope.